thinking about the variation in chromosome number as one type of chromosome mutation that can occur, we can compare individuals that have an abnormal set or number of chromosomes to individuals that have the wild type number. So euploid individuals, U meaning true, refers to a normal wild type state where the individual has the wild type number of chromosomes and copies of chromosomes. So in humans, we have 46 chromosomes. The euploid state is then 46 with 2 copies of each of our homologous chromosomes, one from the mom, one from the dad. An aneuploid individual is a state when the individual loses or gains one or more of individual chromosomes, but not the complete set. So in contrast to polyploidy, where we have a complete additional copy of all of the homologous chromosomes, an aneuploid is a case where 1 or 2 or a small number of chromosomes are different from the wild type euploid state. Aneuploids typically result from non-disjunction events that occur during cell division. So non-disjunction is a process that we'll talk about in a little more detail in a couple of slides. But basically we're talking about the separation of either homologous chromosomes or of the sister chromatids that are joined at the centromere during meiosis. And so we have an example of an aneuploid that's indicated here. An individual with Turner syndrome has 45 chromosomes as opposed to the euploid number of 46. And Turner syndrome is specifically due to individuals that only have one of the X chromosomes. So one of the other chromosomes, the X or the Y, is missing. And this aneuploid state is specifically due to the loss of one of those X chromosomes. Aneuploidy can also lead to the gain of a chromosome. So we see this as a well-known example in trisomy 21 or Down syndrome. This is the only example of trisomy in humans that are viable that involves a non-sex chromosome. This specifically leads to the characteristics that we associate with individuals with Down syndrome, and this is due to overexpression of those genes that are found on chromosome 21. So we have a normal karyotype of an individual here except for chromosome 21. It's a relatively small chromosome. Normal wild type individuals will have 2 copies, one paternal, one maternal copy of this chromosome. Trisomy refers to the fact that this aneuploid individual has an extra copy of chromosome 21. And because typically we have genes that are expressed from alleles present on the paternal and the maternal copy of chromosome 21, when you have an extra copy, those alleles are also expressed, leading to an additional dose of these 200 to 300 protein coding genes. These genes have a wide range of effects. So when we have overexpression, we see a wide range of effects. Things that are impacted include immune system function, heart defects, and skeletal malformation. So these individuals have both phenotypic effects of having an extra chromosome 21. They also have physiological effects, so referring to the functionality of the immune system. And this is primarily due to the fact that they have an extra level of dosage of those proteins that are produced. So if we're thinking about aneuploidy and how it's caused by non-disjunction, we're going back to those stages of meiosis that are kind of the reduction of the number of either homologous chromosomes or the chromatids per cell. So we have non-disjunction that can be caused by the incorrect separation of homologous chromosomes, which happens during anaphase 1, or of the sister chromatids that occurs during anaphase 2. So we have both of those cases demonstrated here in this figure. So starting over here on the left-hand side, we have our homologous chromosomes that go through a first division non-disjunction event. So during anaphase, we would expect one homologous chromosome to go to one daughter cell and the other homologous chromosome to go to the other. So you would expect to see one chromosome over here, one chromosome over here. When non-disjunction occurs, both of these homologous chromosomes stay in their pair and they both end up in one of these daughter cells. The other daughter cell lacks the homologous chromosome. So we can kind of compare what's happening in this first division non-disjunction with what normally happens. So this is what's being shown over on the right-hand side. We have the same homologous pair. Normal disjunction occurs. So during anaphase 1, these homologous chromosomes separate. So one version, one parental copy goes over here, one parental copy goes over here. And we have our first level of meiotic cell division. So here we're looking at the result of telophase 1. Then we have the second round of meiotic division. We've got our non-disjuncted kind of offspring cells from meiosis 1 undergoing meiosis 2. So here again we have the separation of the sister chromatids, and this would lead to two daughter cells that have two each sister chromatids coming from those two homologous chromosomes. So they have twice as many copies of that chromosome, whereas the descendant cells from the daughter cell that didn't receive any of these homologous chromosomes during that first division have nothing to divide. So there's no version of this homologous chromosome over here that could divide into the two sister chromatids. And so while this individual might have other homologous chromosomes that aren't shown that undergo the second meiotic division, they won't have a representation of this particular set of homologous chromosomes over here. So when we look at the gametes, half of the gametes have twice as many copies of this particular chromosome, half of the gametes have no copies. So we can compare this to the situation where we started off normally. So the first meiotic division went well. We have our homologous chromosomes separated into the two daughter cells. Normal disjunction will occur from one of these daughter cells. So we have a gamete that has one sister chromatid each coming from this homologous chromosome. When we have non-disjunction that occurs in the second level division, so anaphase 2, these sister chromatids don't necessarily separate into either of the two daughter cells that are produced from that progenitor cell. So we have sister chromatid 1, sister chromatid 2, both ending up in one of these gametes, again leading to twice as much of that copy of that material here and no copy of that chromosome in the fourth gamete that would be produced. So we have our first division non-disjunction, which is due to the incorrect separation of homologous chromosomes, happens in the first phase of meiosis. And then we have the second division non-disjunction event, which leads to the incorrect separation of sister chromatids during anaphase 2. And in both cases, we have some number of gametes that are produced that have twice as many copies of this homologous chromosome, and some gametes that have no copies of that homologous chromosome. This last row shows what happens when you would have the fertilization event. So a haploid gamete coming from a different individual. If we imagine that this is spermatogenesis, all of these gametes that are produced are sperm. We have an egg cell that would be fertilized by each of these different types of gametes. So over here with our first division non-disjunction, we ended up with two gametes that have twice as much of that chromosome and two gametes that have no copies of that chromosome. And so out of the 4 potential offspring that could be produced with a normal egg cell, we have 2 individuals or half of our offspring that would be likely to be trisomic, so have 3 copies of a particular chromosome, and half of the offspring would be monosomic, so they'd only have 1 copy of that homologous chromosome. And we can compare that to the expected outcome when you have non-disjunction of the sister chromatids, so during the second level of division. Again, you have a normal parent haploid gamete, so maybe this is an egg cell. We have a couple of offspring that are potentially normal, so they have 1 maternal and 1 paternal copy of that homologous chromosome. We have 1 individual that could be trisomic, so 1 maternal copy and 2 paternal copies of that chromosome. And then, like we had in the first division non-disjunction example, the only copy this individual receives is from the maternal parent. There's no paternal version of that homologous chromosome here. Okay, so aneuploidy gets us to differences in the number of whole chromosomes that are present in an individual. We can also have mutations that lead to changes in the chromosome structure itself, and there are kind of four major classes of chromosome mutations that lead to differences in the structure or the content that is present on each of our chromosomes. So these include deletions, duplications, inversions, and translocations. And the different types of mutations are shown in this figure. So we'll walk through each of these one by one. So thinking about chromosome deletions, this is very simply part of the chromosome is missing. So during crossing over, maybe during the DNA replication, something went wrong and led to the loss of genetic material. This is unlikely to be fixed or revert back to the wild type state because that missing piece is essentially lost. It's not anywhere else in the genome, it's just missing, it's gone missing. These chromosome deletions can occur anywhere on the chromosome. They might happen at the two different ends, at the telomeres. We might also see deletions that occur in the middle, which is what's shown in this image. So here we're looking at different genes, A, B, C, D, E, F, and G. When we have a deletion, we're showing a deletion that's occurred that leads to the loss of this gene D. So we've got A, B, C, and then we move right to E, F, and G. Typically, the portion that retains the centromere is the portion that's going to be kept after a deletion event. So in this case, we lost a relatively small portion. If that portion was larger, it might be genes C, B, and A. We would still retain the portion that has the centromere because this is, again, important for mitotic cell division. We have to have that centromere in order for our homologous chromosomes to match up and align and go through mitotic cell divisions. Duplications occur when we have a portion of the chromosome that has been doubled or duplicated. This is something that can be caused through unequal crossing over during synapsis. So if we have our two homologous chromosomes here, and we have, for example, a repeat region, that makes it a little bit ambiguous to understand where these non-sister chromatids from the two homologous chromosomes should precisely match up. And if this happens, this can lead to a slight mismatch in how this crossing over event occurs, leading us to, instead of four identically structured chromatids, we have one chromatid that's not impacted from the paternal copy, one chromatid that's not impacted from the maternal copy, and then these chromatids 2 and 3 are examples where we've lost a little bit of information or we've gained a little bit of information due to the duplication of this C gene here. And so if we think about how these different chromatids could make their way, for example, into the gametes, you can start to see how this duplication event could lead to individual that receives chromatid 3 having too much of gene C, individuals that receive chromatid 2 having a deletion event, essentially. They'd have not enough of the C gene product produced potentially. So we can have duplication consequences that have a negative effect on the phenotype of the individual, but duplications can also have an important evolutionary effect that doesn't necessarily have to be negative. Duplications are why we have lots of copies of our RNA genes, for example, or we have genes that have multiple genes or families that have multiple genes that belong to them that have slightly different functions. So for example, if you have a gene duplication event as a result of a chromosome duplication event, the original gene is preserved and so it can carry out its necessary function. The copy of that gene is not as necessary for the survival of the individual, and so mutations can accumulate in those copied genes, allowing them to take on new function. And this is something that we've seen be the case, for example, in proteins that have multiple subunits. So hemoglobin is a protein that is the combination of products that are produced by multiple genes. Duplication events are one of the reasons why these types of proteins can evolve. All right. A third type of chromosome mutation are the inversions. This is an example of a case where the segment of DNA or the segment of the chromosome is cut out and then reinserted backwards. So essentially we have a cut that occurs somewhere on the chromosome. Again, we have genes A, B, C, D, E, F, and G. We have an inversion that cut out the section between A and B through to D and E. This BCD region was flipped around and then stuck back in that same spot. And so now we have a new order of genes. We have A, D, C, B instead of what we started with. There are two major types of chromosome inversions. We can have paracentric, which does not include the centromere, and pericentric, which does include the centromere. This is an example of a paracentric inversion because this inversion, B, C, and D, occurs outside of the centromere. If it involved E and F, that would be a pericentric inversion. These inversions that occur on chromosomes don't necessarily lead to the loss or gain of genetic information, so we're not really losing necessary genes or anything like that. We're not increasing the number of genes that could potentially produce a product, but we do have a different order of the genes along the chromosome. So we covered genetic mapping, which allows us to understand the order of genes when we do a 3-point test cross. This has some relevance for understanding how genes might be regulated together. So if we think about genes that are right next to one another on a chromosome, promoters may influence the expression of more than one gene that are sequentially located on a chromosome. We see this in bacteria a lot of times. So there's a potential effect of the position of those genes that could lead to a change in gene expression due to the location change. If this inversion process happens in such a way that the coding regions of two genes come in contact with one another, so they essentially went from two genes that are separated by some space to two genes that are separated by no space, there's a possibility that they'll be transcribed together as well. And so this could potentially produce a protein that is now a combination of two genes or parts of two genes. And this novel chimeric protein could take on potentially gain-of-function effects, so they might have a negative consequence in the cell, but maybe they have a slightly different effect that is essentially neutral or slightly positive. So this is another opportunity for evolved changes to occur.